So in this video we're going to talk about light-dependent reactions, chloroplasts, and light-independent reactions, or you might see it referred to as dark reactions. So the general structure of a chloroplast, it's broken up into three main parts. So the outer envelope, which is the outermost layer of our chloroplast, the inner envelope, which is the inner membrane of our chloroplasts, and the internal membrane, or the thylakoid membrane. So the one that's really important to us here is the thylakoid membrane, or the internal membrane. So if we were to have a thylakoid right here, the inside of our thylakoid is referred to as the lumen, and the outside in all of this area is referred to as the stroma. Also, we have a bunch of thylakoids stacked in flattened pancake form, one on top of the other. We refer to that as grana. So our light-dependent reactions are going to happen embedded into the thylakoid membrane. So these reactions directly use light energy or photons to power the reactions. So we have three complexes that we talk about in our light-dependent reactions. We have photosystem 2, which is the first one that we'll talk about, photosystem 1, and cytochrome B6F. Cytochrome B6F is a lot like our cytochrome C in the electron transport chain. We'll talk about that in a little bit. So our light-dependent reactions are kind of the opposite of what we do in the electron transport chain. At the end of the electron transport chain, we're reducing water, but at the very beginning of our light-dependent reactions here, we're going to be oxidizing water. So we're going to use two water molecules. So two water molecules are going to hold a total of 4 electrons. So just keep that in mind. So we're going to be, throughout these reactions, we're going to be talking about 4 electrons. We'll try to keep it on the right track. But we're doing 2 water molecules because we want to release 1 molecule of oxygen. So we'll be splitting these water molecules by a combination of using this complex, which is called the oxygen evolving complex, and photolysis, which is using light energy from photons to lyse this water molecule. So after these electrons, which in all reality, these electrons are going to go through the system one at a time, but we're going to be talking about them going through in groups of 2. But after these electrons are removed from the water molecules, the protons from the water molecules are going to be released into the lumen of our thylakoid. So we're going to have 4 protons being released into the lumen from these 2 water molecules. So keep an eye on that because it's going to be important in the final count. So after this, our electrons will be transferred through the oxygen evolving complex, and they're going to stay at a chlorophyll dimer called P680. So one thing to note is in its unreduced form, P680 is referred to as P680 plus, and this is the strongest oxidizing agent known, the strongest biological oxidizing agent known. And when P680 is reduced by electrons, by these electrons from water, we refer to it as P680 star. Okay, so P680 is the first part of our reaction center of our photosystem 2. So this light harvesting part is the light harvesting complex of our whole photosystem, and P680 is the first part of the reaction center, which the reaction center is this part in white. So our electrons are going to sit at P680, now P680 star, and they're going to wait there to be excited due to these photons. So these photons are going to travel through our light harvesting complex, which is these purple parts, so light harvesting complex 2 for photosystem 2. And it's going to, these photons are going to be absorbed by pigment molecules, specifically pigment antenna molecules. And these pigment antenna molecules are going to transfer the energy from these photons down our light harvesting complex, and they're going to transfer this energy all the way down to our reaction center to excite the electrons in P680 star. So for every electron we're talking about, we're going to say that it's excited by a single photon. So to excite 4 electrons at our photosystem 2, we're going to use 4 photons. So these 4 photons are going to excite each one of our electrons to a higher electron state, a higher energy state. So we call that the excited state. The original state was at ground state. So now we've raised our electrons to a higher energy state above what they had when they were in the water molecule. So we're going to raise the electrons to a higher energy state, and they're going to be accepted by a primary electron acceptor at our photosystem 2. So that primary electron acceptor in photosystem 2 is called phaophyton. So just like when P680 plus was reduced, it got a name. Phaophyton will be referred to as phao negative when it's reduced. So from phaophyton, our electrons can be transferred to a few electron acceptors, or a few electron carriers, electron cofactors. So the first one we're going to transfer them to is going to be called PQA, or plastoquinone A. And if you remember, plastoquinone sounds a lot like ubiquinone. So these are really similar molecules. So from PQA, we're going to transfer it to a molecule called PQB. And PQB is going to be our molecule that we're going to refer to as plastoquinol. So just like ubiquinol, or just like ubiquinone to ubiquinol, when we reduce a plastoquinone, it's going to become plastoquinol. So plastoquinol is going to travel either on the outside of our thylakoid membrane on this stroma side, or it's going to go really close to the stroma side. And while it's traveling through to our next complex, our cytochrome B6F, it's going to pick up 2 hydrogen molecules due to the 2 electrons it's going to be carrying. So at that point, we're going to refer to it as PQH2, or plastoquinol that's carrying 2 protons. And remember, since we said that we are using 2 water molecules, we're going to have this occurring twice. So we're going to have 2 PQH2 molecules for what we're talking about. So from our PQH2, our carrying molecule is going to travel through our cytochrome BCF, and it's going to hand off the electrons from PQH2 to our cytochrome B6F. So as it hands off the electrons from PQH2, it's going to lose those protons that it was carrying. So now PQH2 is back inside the thylakoid, and then our PQ, now PQB, is going to travel back up to the photosystem 2 and repeat the process. So as these electrons are handed off to our cytochrome B6F, just like our cytochrome BC1 in the electron transport chain, this cytochrome B6F is a proton pump. So not only are 2 protons going to be dropped in from PQH2 dropping them off, but as these electrons travel through cytochrome B6F, 2 more protons are going to be pumped in. Right, so remember we said we're doing this twice. We're going to have 2 PQH2 molecules, so we're going to do this whole process twice. So we're going to have 2 pumped in for 1 PQH2, then it means we'd have 2 more pumped in for another PQH2. And we had 2 protons that are dropped off for 1 molecule of PH2, so we're going to have 2 more dropped off for another. So this is going to give us a grand total of 12 protons being brought into the lumen by these reactions. So we had 4, I'm just going to clear up some space. So we had 4 protons from water, 4 by the pumping of cytochrome B6F, and 4 by PQH2 dropping off protons into the lumen. Okay, so from cytochrome B6F, we're going to take our electrons to our photosystem 1. So the electron cofactor, the electron carrier for that, is called plastocyanin. And this is just the carrier from cytochrome B6F to photosystem 1. And our plastocyanin is an internal peripheral protein, which means it's a protein that travels on the outside of our plasma or our thylakoid internal membrane. So our plastocyanin is going to hand off our electrons to the other pigment molecule, the first molecule of the reaction center for photosystem 1, and that is called P700. So just like P680, P700 when it is oxidized is referred to as P700 plus, and P700 when it's reduced is P700 star. So now our electrons are going to sit at P700 until they're excited by 4 more protons, because remember we said we're talking about 4 electrons, so we're going to need 4 more protons to excite them to an excited state to be caught by the primary electron acceptor of P700. So that electron acceptor is called A sub 0. So when A sub 0 is reduced, we refer to it as A sub 0 minus. So from A sub 0, we're going to transfer our electrons to a few cofactors. So first it goes to Fx, then it travels through Fb and Fa, and then our electrons finish up at the cofactor called ferredoxin. So ferredoxin is just an electron cofactor that will take our electrons to the final resting place, which is referred to as ferredoxin NADP plus reductase. So just to recap, our electrons go from A0 negative to Fx. They travel through Fb and Fa. Then they are transferred to ferredoxin, which will then transfer our electrons to the ferredoxin NADP plus reductase enzyme. This enzyme is located on the outside of our thylakoid membrane. So this enzyme is located on the stromal side, or the stroma side of our thylakoid membrane. So for every 2 electrons that travel to our ferredoxin NADP plus reductase, which we said we're talking about 4 here, so it's right there. For every 2 electrons that travel to this enzyme are going to be responsible for creating 1 molecule of NADPH. So NADP plus 2 electrons plus our hydrogen equals 1 NADPH. So for the creation of 1 molecule of O2, the expenditure of 2 water molecules will be making a total of 2 NADPH. So from this process we've created 2 really high energy molecules for spinning energy in the Calvin cycle, and we've created a proton gradient. So let's just do a grand total of everything we've done. So we brought in 12 protons into the lumen. So that was 4 from water, 4 from cytochrome B6F, and 4 from PQH2, dropping them off into the lumen. We've created 2 molecules of NADPH. And now we are ready to use these molecules of NADPH, directly to the Calvin cycle. And these protons, and our protons are going to be used to directly power ATP synthase. One other important thing is we used 8 photons here, 1 for each electron at both our photosystems. So we used 4 photons at photosystem 2 and 4 photons at photosystem 1. Okay, and the last thing you need to know about photosynthesis is a little bit about the Calvin cycle. So you need to know that we're going to be using 6 molecules of CO2, that being, or the reason being is because 6 molecules of CO2, 6 carbons will equal 1 glucose. So for 6 molecules of CO2, we're going to be using 6 molecules of RUBP, and RUBP stands for ribulose bisphosphate. So ribulose bisphosphate is a 5-carbon molecule. So we're going to be starting off our RUBP, I'll use different color. Our RUBP alone for 6 molecules is equal to 30 carbon. So our 6 CO2 are added on to our 6 RUBP by the enzyme Rubisco, the most abundant enzyme in biology. So Rubisco catalyzes the reaction of adding on 6 CO2 to our 6 RUBP. So immediately when these two are added, they are incredibly unstable and they fall apart, or they split apart into 12 separate molecules of PGA. 12 PGA, or 3-phosphoglyceraldehyde. So we started off with 6 molecules of RUBP, or ribulose bisphosphate, and we added on 6 CO2 by Rubisco, which is ribulose bisphosphate carboxylase. So that kind of makes sense. We've carboxylated these RUBP molecules, and they've split apart into 12 PGA molecules. So just to keep our carbon count, our 12 PGA molecules are equal to 36 carbon. So that would be 3 carbon for each PGA molecule. So from this part of the cycle, all you need to know is that this is where we're going to be expending all of our energy molecules that we made in the light-dependent reactions and from ATP synthase. So we're going to put in 12 molecules of ATP, and we're going to put in 12 molecules of NADPH. So one thing to remember is, we made NADPH and we let it go into the stroma. So we always let our energy-carrying molecules go into the area that they're going to be interacting with whatever cycle we're talking about. So we can be for sure that this cycle is happening in the stroma. So after we spend our 12 NADPH and our 12 ATP, our molecule is now going to be referred to as GAP. So we're going to have 12 GAP molecules, or glyceraldehyde 3-phosphate. So from here, we're going to export 2 of our GAP molecules. So 2 GAP molecules is the same carbon formation as PGA, so that would be, we're losing 6 carbon, and we are left with 10 GAP, which will leave us with 30 carbon. So our 10 GAP, each one being 3 carbon, leaves us with 30 carbon left. So our 2-GAP, our 2 glyceraldehyde 3-phosphates, will go off to make some sort of sugar. It could be glucose, lactose, whatever. And what we want to do is we want to turn our 10 GAP back into our original 6 RUBP. So to do this, we're going to have to spend 6 more molecules of ATP. So we spend 10 molecules of ATP, we rephosphorylate all of our GAP molecules, and they reform our 6 RUBP. So we've gone from our 10 GAP, which is 30 carbon, back to our 6 RUBP, which we said is also 30 carbon. So this whole cycle, we spend 18 molecules of ATP and 12 molecules of NADPH. We do all of this to make 2 molecules of GAP, which could then go on to make whatever sugars. So going back to our light-dependent reactions, what would happen if I didn't want to make NADPH molecules? What if I just wanted to make ATP? Well, what we described here in the light-dependent reactions is referred to as the non-cyclic or linear photophosphorylation. Photophosphorylation being a term used to describe the phosphorylation of ATP driven by this light energy. So what if I didn't want to make NADPH, but I just wanted to make a proton gradient. I just wanted to make ATP. So everything would be normal up until we got to ferredoxin. So our electrons get to ferredoxin, and instead of handing them off to ferredoxin NADP plus reductase, we're just going to take our electrons right back to cytochrome B6F. And when we take our electrons back to cytochrome B6F, it's going to cause more protons to be pumped in. So if we were taking 2 electrons from ferredoxin back through cytochrome B6F, it would cause 2 more protons to be pumped in. Then our ferredoxin will return to where it was, and our cytochrome B6F will hand off the electrons to plastocyanin, just like it did last time. And we'll just keep doing this to make this proton gradient that will power our ATP synthase. So this is referred to as cyclic photophosphorylation. So our earlier process was referred to as non-cyclic photophosphorylation. That was linear. But now we want to go in a cycle between ferredoxin and cytochrome B6F. So one last thing to note is when we are doing linear photophosphorylation, we are, at photosystem 2, raising our electrons to a higher energy state than water. So this is water's state for electrons. We're raising it up to a mid-level. And as our electrons travel through cytochrome B6F, just like an electron transport chain, they're going to be losing energy. So when they get to P700, they're going to be in a very low energy form. So that's why we use photons to raise them back up to a higher excited energy state. And as they travel through FX, FB, and A, the ferredoxin, they're going to be reduced a little bit. But when they get to ferredoxin dash NADP plus reductase, they're going to be at the perfect energy level to reduce our NADPH molecules.